Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall provide an introduction to programmed cell death before we talk about some of the programmed cell death pathways in future videos. As we discussed in the last video, a cell can die either by accidental cell death, ACD, or by regulated cell death, also called programmed cell death, RCD or PCD. In this as well as future videos, we shall focus on programmed cell death, and this is cell death that is regulated, is under genetic control, unlike what we see in accidental cell death. Accidental cell death, the cell dies mostly by necrosis, where the cell is broken, lysis, and there is inflammation. Programmed cell death can happen in many different ways, such as apoptosis, and there are several other PCD programs that we'll mention in this as well as future videos. ACD occurs in pathology, while programmed cell death occurs both in health as well as in diseases. As explained in the previous video, programmed cell death maintains tissue homeostasis by removing non-functional cells or damaged cells or harmful cells in health as well as in pathology. It is estimated that the whole body weight of cells undergo regulated or programmed cell death in 1 year. PCD is extremely important for human survival. There have been many different types of programmed cell death that have been reported. The literature on programmed cell death is very extensive. Several of these programs were discovered only very recently, and therefore we still do not know much about them. Some of this literature actually is very confusing and even sometimes contradictory. So we'll try to keep this material on PCD in this video as well as in future videos as simple as possible, and we'll try to focus mainly on the well-established aspects of PCD as well as the important concepts as they relate to medicine. One important question that comes to mind when we look at the literature on programmed cell death is there are so many different pathways that have been reported. Why does a cell choose a certain PCD pathway? There are several factors. They can be quite complex, but let's look at a few examples. One factor is the type of stress that the cell faces. For example, if a cell is infected with a virus, then it can undergo apoptosis, or it could undergo necroptosis, or even pyroptosis. If a cell has excess iron, then it will undergo the programmed cell death called ferroptosis. Similarly, if the cell has excess copper, then it will undergo a programmed cell death that's called cuproptosis. If there is an increased intracellular pH in the cell, then the cell can die by a PCD called alkalyptosis. The second aspect is benefit to humans. When we look at the whole picture, now cells basically need to survive so that the human can survive. So when a cell dies by programmed cell death, there must be some sort of benefit to the whole organism, in this case the human being. Let's consider once again a cell that is infected with a virus. So in this case, the stress once again is the viral infection. Now the cell is first going to try to fight off the virus. It's going to prevent propagation of the virus. Remember, viruses require the cell DNA as well as protein synthesis machinery to propagate itself. So the cell is going to try to stop the viruses from multiplying. And if it succeeds, then the cell is going to initiate a pro-survival program, especially if the cell cannot be replaced, such as a neuron. And then the cell will also release or may also release pro-inflammatory materials to warn the neighboring cells as well as the rest of the body of the impending danger with the viruses in the cell. Now, if the cell realizes the virus is winning and the viral propagation cannot be stopped, then the cell is going to choose one of these programs, and it might choose apoptosis, where there is no inflammation, or it may decide to go into necroptosis or pyroptosis, in which case there is inflammation that will help warn and stop the viruses from affecting other cells in the body. In regular physiology, during tissue formation, maintenance, and repair, examples we saw in the earlier video, such as sculpting tissues, maintaining organ size and shape, removing excess neurons that do not make successful connections during development, the menstrual cycle, where the old uterine lining is replaced to make a new one, removal of autoreactive T cells in the thymus during development. In all these cases, these cells these excess cells need to die, so that's done by programmed cell death. But in this case, we don't want inflammation, so the cell will therefore choose apoptosis where there is no inflammation. And this is important because remember, inflammation not only will affect the stress cell, but it can also affect the healthy cells. Another factor that that affects what the cell is going to use as its PCD pathway is the cellular environment. Coming back to the example of a virus-infected cell, this cell may choose apoptosis to limit inflammation, which can harm neighboring cells. And in case the virus produces proteins that inhibit apoptosis, and we know viruses do that, they are very smart, they can actually produce proteins to stop apoptosis. So in that case, the cell is going to use another pathway. For example, necroptosis, where there is inflammation, to warn the body of the smart virus that is producing proteins to stop its apoptotic pathway. So now the cell uses necroptosis with inflammation. Or the cell may, instead of using necroptosis, may use pyroptosis. And that's because pyroptosis is much faster than apoptosis. So the faster the cell dies, the less the viruses will be able to propagate and affect neighboring tissues. We'll come across several other factors as we discuss PCD in future videos. A few words about the medical aspects of programmed cell death. Programmed cell death, as we shall see in future videos, is very closely associated with inflammation. Now, inflammation is an extremely important process in pathology. So those of you who will be taking or have taken pathology, you probably know how much time is spent on studying inflammation. Now, too little cell death can lead to this dreaded disease, cancer. On the other hand, too much cell death can cause degenerative diseases such as the neurodegenerative diseases, Alzheimer's, Parkinson's, Huntington's, and so on. Since cell death is related to diseases, it is important in pharmacology. For example, the apoptosis inhibitor, venetoclax, is used for treatment of certain hematological malignancies. So we can develop new drugs if we know where and when a certain program cell, that pathway, is activated in a certain human disease. In future videos, to avoid confusion and to explain well-established concepts of PCD, and also due to their medical importance, we will focus only on a few PCD pathways, especially apoptosis, necroptosis, pyroptosis, and a little bit on ferroptosis. The rest will probably provide a quick overview. Before we discuss the actual PCD pathways, in the next video we are going to talk a little bit about these proteins, caspases, enzymes called caspases, as well as dead domain proteins. So that's a little bit of an introduction on PCD. Thank you very much for watching. Goodbye and have a wonderful day, and hopefully we'll see you in the next video.